I hopefully I'm audible and my screen is visible. Starting in a minute.
Okay, sir.
I'm audible, right?
Yes, sir.
Yes, sir.
Now, if anyone of you can just unmute and say, okay, so we'll be starting with today's live session for the week one. So we'll continue all in this live session for the course and prepare for introduction to protegenomics. So if you all, so this is the live session and it will be an interactive session. So if you all just get any questions or anything you want to ask, you can interrupt and ask. So this is also been
Sir, your voice is
appreciate
breaking.
that is anyone saying anything?
your voice is breaking.
I'm not able to hear. I don't know why. Can you just type in chat or something?
Hello, now am I audible now?
Yes, sir.
Okay, thank you.
Yes, sir.
Yeah, so I think there were some issues. Sorry for those things. So today is the first week one live session for the Introduction to Protegenomics course. So it is also being live streamed at YouTube. I think the YouTube link will be provided by the NPTEL team. So I will start by brief introducing myself. So I am Ankit Haldar. I am a MTech PhD dual degree in the proteomics lab, IIT Bombay. I am in currently in the third year of my study. So I'll be here in this course. I'll giving a little bit more concept of what you have all have done in the lecture, along with some of the things and extra computational tools, and how we can go ahead and perform those things that have been told in the lecture. So I'll start here by going with the proteogenomics. So hopefully you all have gone through the lecture for this week one. So here I'll give you a little bit of description or the definition of what you can understand by the proteogenomics. So you all have might have seen in the lecture that it is a combining why genomics or proteomics, why both are required, but how and where we need the integration of it, or how we can do this integration, so we will be able to understand the biology between various diseases well. So also I would request you can, if you all have any question or any doubt or anything, you can interrupt and ask, since it is an interaction session, live interaction session. So here we go by the definition of the proteogenomics. So it requires both genomics aspect as well as the proteomics aspect. So for instance here, if we can take a tumor sample and we take out DNA and perform whole genome sequencing and whole exome sequencing. So you all, I hope you all are familiar with the terminal of whole genome and whole exome sequencing. So whole genome means we sequence the whole genome from an organism's tumor tissue, or in, whereas exome sequencing is that part of the genome sequence which codes for further RNA or the protein sequencing of those. So now from here what we can get is that we know that we can identify somatic mutations, copy number variations, different kinds of microsatellites, and etc etc. Similarly, from RNA sequence we can identify the gene expression, novel splice variants, mRNA expression, etc etc. Now, and from the protein by the mass spectrometry, I think you will get to all know in a detailed way in the when the proteomics aspect will come in the lectures. We can identify different peptides. Peptides in the sense of from the proteins, when we digest and get the peptides, we get identify those peptides, and from where we identify what proteins are there in our sample. Now when we perform a proteomics based experiment, when it was performed in a data dependent acquisition mode, we require a database to identify those peptides. That is corresponds to the organism's database, known database. We identify those peptides with our invasive fingerprints. So now where this integration and all, from where the definition can come is that, since we generally identify our protein database from a reference database that is already present in different websites like Uniprot or different references databases, but that always does not account for an individual patient specific mutations or patient specific any differences, that doesn't account for, and we miss those information. So when we get a DNA and RNA from the same patient and we get to know those sequences, and if we can prepare a customized database, we can see that a customized database, so then all the mutational information for those patients specific we get to know. And when we can identify those peptides or the protein data what we get, if we identify with that database, we will get to know about the mutated peptides or the mutation protein, or that genomic level whatever those sequences have been mutated, we can get those sample specific or patient specific peptide sequences, which can help us in further analysis is biomarker discovery, disease prediction, and the novel pathways. So here we are getting a more about a customized, a patient specific database, a patient specific, we can get capture the information. Hence the proteogenomics approach is a more powerful approach, where a patient specific thing we can acquire, we can identify. As we all know that the tumor is itself is a heterogeneity, and in that patient wise there are many heterogeneity and subclasses within the groups. So here we can easily capture the full information by using this approach. So this is what our definition of proteogenomics approach can be told in a way. So now moving on, so you all have, I think, if you all have heard the lecture of Dr. Kelly V. Ruggles, and also if you have gone through, and Dr. Money, Dr. Money, and so these groups are those who are mostly taking in the clinical proteomic CPTAC papers, and who are working in the proteogenomics field. So they have this is a excellent review article from that group, which have expanded the meaning of the proteogenomics approach, and how what applications can we do from the proteogenomics approaches. So you all can go and read this paper to understand much more. So here we can see that this DNA, as I have said, whole genome and whole sequence. So here we need to perform two alignment to a reference genome, because for any genomic sequence we need to have a reference genome sequence. So our reference genome sequence is generally like HG19, HG38, which are present, that can be used. Also it can be taken from the same patient, like if we want more specific analysis or more specific requirement, then a tumor DNA from a tumor sample, and the DNA, if it is happening in a tissue the tumor has happened, then you can take out the DNA from the PBMCs or the blood cells from the same patient, and that you can take as a reference genome which is not mutated and the tumor tissue is mutated, and then you can align it. And then is the variant calling. So variants, you all know that there are many kinds of variants: single nucleotide variants, SNVs, which are those which occurs at a single nucleotide level, that can be further classified into missense or nonsense or silent mutations. Then indels, then the gene fusions, all these you can go from RNA-seq. similarly from the peptide. And finally you need to have a customized sequence database. Here is the catch, here is the trick to get to that customized sequence and then identify those peptides. And finally there can be mutation analysis, genome reannotation and all. Second thing which the application, if you can see that here you can subdivided, generally mostly the cancers and the tumors are subdivided into various, World Health Organization classifies it into various grades or various subtypes. So you can here further you can check that within that subgroup, for example, a subgroup of a tumor, like for example in GBM, a brain tumor, in the glioblastoma tumors only are there existing any different subgroups, or in the different grades of glioma are there corresponding to it, or with this approach you are getting a different classes of these mutations, different subclasses, if they are, can be divided into any further subclasses or not. Then predictive modeling, we can predict, diagnose, you can prognosis how can a drug react. For example, a drug, a drug targets a certain protein and it is being administered, but when you go through proteogenomics approaches you found out that those mutation is not playing a role for a particular patient in that case, and then if you are adding, giving the drug, that might not act, other or one some else drug can be added and can act. Third is the pathway and the protein interaction and the gene enrichment and the functional relationship that is required to understand the different novel pathways or different novel interactions that are taking place. So this is the further modeling of applications of this proteogenomics approaches. Another thing is that required is that the correlations, like the mRNA protein correlations, mRNA to protein expression. Okay, so how means all the mRNA at the level of changes in the mRNA level, are they being affected in the protein level or not? So it can be positively correlated or the negatively correlated. So what is that expression correlation? Hence, any one approach we cannot see the whole picture. So hence the both the approach is required. Then copy number alteration to the expression data, how much it is changing. DNA expression variation to the expression data, or the miRNA or the different kinds of RNA, that regulation how they are being functional. So then you can see that the DNA variation to the PTM size, that the PTMs are also being regulated. So various kinase pathways or altered pathways generally gets altered during the tumor or cancer pathways. So how much they are getting affected. So now let us look at a case study in the sense that the research article. So here I have thought that we will be looking at a case study every week to a small part, that how, so that you can get an idea that how this technical knowledge is being actually implemented in the helping in the research, and how it is being getting published, so that you can also get the confidence and understand how to analyze and interpret this kind of data. So here you can see that this has been one of the CPTech papers of the proteogenomic characterization of human colon and rectal cancer. So here we can see that what we tell theoretically, that what is the effect of the changes in the genetic level, in the protein level, and how it is actually done. So if you can see that it is a they have taken from TCGA. So TCGA is the Cancer Genome Atlas, and all those genetic changes are been reported and have been done and reported here. Cosmic is one of the database which contains a catalog of somatic mutations in cancer, and DBSNP is a SNP database. So if you all know, so here they have identified the single amino acid variants from their data, from the somatic mutation, from Cosmic, from DBSNP, and identified some of those mutations. Now, how they have tried to identify that their effects on the protein abundance. So, are these mutations that have been taken have been identified? Are they also being affected at the protein level? For this, they have taken the help of statistical tests like Kolmogorov-Smirnov test and chi-square test to identify that whether what is the effect of the proteins, the single amino acid variants at the protein level. Are they affecting or not, or what is the case? So how they did it? So here you can see that impact of SNVs on protein expression. So on non-synonymous protein coding, so we all know that the synonymous protein coding will not change at the protein level generally, because it will be remain the same. The amino acid will remain the same. Where for their existence, for each SNV in one of the three categories, that the cosmic, DBSNP, and the somatic mutations, if the SNV containing gene is included in those genes and with sufficient spectral count for comparative comparison across samples. So if that particular gene is included in the proteomics data and it has the sufficient count, because all the proteomics data always doesn't get detected. So if it is contains those spectral count, so then the SNV on protein expression by comparing protein expression of the gene in the SNV containing sample and without the containing the SNV samples. So the protein expression that was found, the nature of the expression of the protein that is present on the SNV containing samples and without the SNV containing samples, using the simple formula like EXP is the expression containing the SNV. So the protein expression what we get from the mass spec data, so what is that expression level in the containing the SNV, and the median of the non samples which are not containing any SNVs, and the mean absolute deviation of the expression containing the all the samples without SNV. Okay, so this is how mm, they have calculated the impact score. And after you calculate the impact score, you can calculate that within two SNV samples, what is the impact for that particular variant on the protein abundance. So hopefully you all got it, how you can identify those effect, how with the help of calculation, with the help of statistics, you can really determine the effect of abundance of those single abundance on the protein variance level. Similarly, correlation between steady state mRNA and protein abundance. To compare those steady state within individual samples, all within a sample have to be comparable. So then you have to think of the what are the measurements, like for example FPKM, or the fragments per kilobase median reads are in the RNA, and the similarly protein expression will have. Then you have to scale it. After scaling, you can find out the correlation coefficient using like Spearman correlation coefficients. So with the normalized protein expression values and with the FPKM values, identify that which are how, what is the correlation between the mRNA and protein, the is it positive correlation or the negative correlation. And protein mRNA and protein variation, so the protein and which you identified using those genes, for those genes you have to identify those expression values for those particular genes which you have the proteins, you have the proteomics data. And then after normalized proteins data and the normalized expression values of the RNA-seq, you can further you can perform a correlation coefficient and identify that what is the abundance. So this is how, if you go and study this paper, so here I've just given a few 2-3 examples. So here the how you can correlate between the mRNA and the protein abundance. For this is just an example of how proteogenomics can help us to understand uh, different methods, different correlations. But further, one can go and download their data perform reanalyze to get a new aspects of thing. Also the thing is that one can perform the customized database and search from these data. One can search their own proteomics data with the database of with the data they have provided to look that how much is the difference between those protein levels and between the patient levels of ours. Or if you search with the simple uniprot database, what are the differences, then you can appreciate that why proteogenomic characterization and proteogenomics is required and such beneficial for everyone. So moving on. So here, if you see the proteogenomics approach, so first one aspect is the sequencing technologies. Like we all know that this has been possible. The emergence of the proteogenomics technologies have been possible with the emergence of the help of the advancement of the sequencing technologies. I think you all have learned about the Illumina sequencing technology, where the huge scaling can scale up could be done very much easily. And then we need to analyze the data and then interpretation of the data, like all possible variants. Again we should need to perform computational screening, and after the screening only we can identify the effect of the mutation, the effect of those at the protein level, at the phenotypic level. So the genotype to phenotype association has been a major challenge and major importance. The realization has been done that need to understand. So for this we need to understand both the aspects very well, the genomics as well as the proteomics technologies, and where we can, wherever we find we should help in complementing and implementing both the techniques together. So the sequencing technologies will give us a huge amount of data, like you know about the paired end sequencing or the single end sequencing, or the sequencing data which we get to know about all the genes. Then we need to identify those genes. We need to have a stringent quality control check of those sequences, whether they satisfy the criteria of passing to further taken to the analysis. Then to align to a reference genome, identify those variants, how those variants are affecting at the proteomic level, identify those mutant peptides, are they conveying to us any useful sequence or any external extra thing that we don't know till now. We convey this those can those peptides, mutant peptides, be used as a for a immunotherapy or for any kind of targeted therapies, or for any novel kind of diagnostic tool or diagnostic assay can be prepared. So all these questions needs to be addressed by after analyzing and after interpretation of those data. And for this computational tools and algorithms play a major role. So with the course of the time, I'll let will also you will know different computational tools that are being employed, different algorithms that are being employed to analyze those data. So we'll try to give example of as much as possible within this course of time. So here if we go, so you can understand that there is a genomic data commons of the National Cancer Institute with the TCG and the CPTAC paper with the CPTAC program launched to access to help the people understand in the all those to publicly access all those data. So once genomic data commons is one of those portal which is used for data repository and sharing. Similarly of the CPTAC thing, and for another is in the of the NIH is the sequence read archive, which contains various sequences for the RNA sequencing data that people deposited here, which can be used for further analysis. So there are different public repositories from where we can download the data and can start analyzing our data. So for this week, so before going on to all those different complex tools and algorithms from what has been described, so here let us first brush up a little bit of what we do in a basic bioinformatics. So we all know the gene bank, that is mostly the comprehensive sources of whatever different IDs present. So for example, the sequencing data, generally we look at the FASTQ format for the DNA sequencing or the RNA sequencing, but all those are a form of the FASTA format, which we all know. I am hopeful that every one of you are aware of it, but still we need to a little bit brush up a little bit. So mostly this remains in the FASTA format, where it starts with a greater than sign which contains the definition, description and the sequences. So so let us try to brush up a little for this week the different tools that we know, because the alignment to a reference genome is nothing but it's like a string matching algorithm, where you match between an algorithm, and the most basic is the uh, blast, what we perform to identify those sequences or identify the uh, polymorphisms at the sequencing level. So if we go and try to identify those, from there we can increase our complexity and we can understand our different computational tools and different computational tools available. So if we go and for your preferences, like, so here it is, and we all know that it contains an accession number. Yeah, so here you can see that by accession number we can identify that which is the, that is human hemoglobin alpha HN is what we are getting, correct. So from here we can see the sequences that is present. This is the sequence, and if we go to the FASTA, so we can get this FASTA sequence. So this is the FASTA sequence. So you all can also do along with it. If you all know also, then also you can just go and try to refresh it. Screen now it's visible, right?
Yes, sir.
Okay. Yeah, so this is the FASTA sequence. Hopefully you all can also get. So here this is the accession number. This is the name of the protein, and this is the sequence. So this is the sequence of the FASTA. Now, if you all go search in emboss secret, so you all can see emboss secret, right? So here, if I just paste the sequence, in any supported format, that is the FASTA format. Now if we change it, that is if we change it into FASTA, and if we go and change it into see, here you can see that FASTQ format, FASTA format, everything is there. And in the GCD sequence format, if you submit it, so see, you can find out that different sequences. So these different sequences have been formed to identify, to help the sequence to in the different tools to recognize those sequences and to identify those sequences. So here we can see that we can change to sequence to any.
Excuse me, sir.
Yes.
So your screen is not visible. You're not sharing your screen.
Okay. Uh, I don't know why it's not getting shared. Actually, it's not letting to share rather than this tab. I don't know why. Uh, okay, let me just paste the sequence here. So with that thing, I think you all can just, if you all can just uh, yeah. So this is in the emboss secret. So you all can, if you all, I don't know, this thing has been recorded in the video. You can check it out how to do it in the emboss secret. So if you just go to the emboss secret, so you can search here. I am writing in the chat. So if you all just type it and paste the sequence in the FASTA format, and you can try to that here it is much straightforward, and you can just change it into the any kind of desired format. Here I have done it in the GCD format of the different. So actually these types of formats are required for the tools to recognize, and here in this way you can perform and change those sequence results. Okay, so moving on. So the sequence alignment. So as we are telling that the sequence alignment, we need to align it to a reference genome. This we need to identify the variants by after aligning the sequences and all. So the thing that came that has been the basics of all this had been to understand the sequence homology, the sequence similarity, and the sequence identity. So the this is the basics for all the different algorithms, like the dot matrix algorithm. Starting from those, people has tried to align the genomic sequences and identify how much they are similar to a particular species, or how much similar, or two species are how much similar to each other based on their genomic sequences. or the sequence identity. Now we'll know that there exists the two kind of alignment. One is the global alignment, which helps to align for the entire genomic sequences, and the local alignment is performed generally over a particular region by region. So we all know that the efficiency of the algorithm and the computational complexities, if we try to align the whole genomic sequences, it will take much more time, it will be a much more tedious thing. Whereas if we try to region by region sequencing, which is much more logical, as well as identification, helps us to reduce the complexity as well as help in identifying those tools in a better way. So this is the concept behind the sequence alignment that has come, and that is extending to such a level that now we can align using different kinds of tool, which we like for the for proper alignment we use like BWA or Bowtie 2 or those tools. But before going on to those tools and how those functions, so let us try to do basic what we all know from R, that is the blast. So that requires so blast is nothing which is a heuristic which contains a whole lot of information. So with if we collect from a bacterial sequence or any kind of sequence, and if we get an unknown sequence, so with what sequences it gets matched, or how we can identify that with which sequence it is identical. So we generally use blast. So it is performs the database searching method. So here what does blast, how does this algorithm of the blast does, so why it is important to understand these basics and their algorithms before going into those complex things, because that these all help us in understanding that on what basis the tools are generally built upon, by extending how it is revolution means how it is evolved, how does the algorithms also evolved, or how does a tool gets evolved, on how what idea, and how further we can improve those tools and algorithms, and how further we can help us reduce the time complexity, because of those big sequences. When you try to align, or if you don't break it down or don't understand that how it can be efficiently improved. So the proteogenomics concept and the approach relies heavily on the different computational tools and algorithms, without which analyzing the data and interpretation of it will become much more difficult, and we will be stranded with no choice of how of understanding uh, how we can interpret those big sequences and the big data. So there are we all know that still for the refreshment, that there are four types of blast that are generally is there. Nucleotide blast, where a nucleotide sequence can be matched with a nucleotide sequence to identify that how much are identical. And again is the blast X, where when you provide a nucleotide sequence, it will translate it into 6-frame translation or the 3-frame translation according to your command, and it will, with the protein database, it will search with the nucleotide. So from the nucleotide sequence, with what protein sequences it will get matched, you can, so if you identify an unknown nucleotide sequence, you can easily find out that with which protein sequences it is getting matched. Again is the T blast N, from the protein sequence. If you sequence an protein, or if you express one kind of protein, and you want to identify that it is from the same nucleotide region or not, so you can perform T blast N, where it will be matched with the translated nucleotide sequence. And another is the protein blast, which you match with the protein. So here generally what we look upon in the blast result is that the percentage identity of the value, how much the maximum number of hits with what we are getting, and the e value. So e value is that the expected value means that the hit is by chance or by or actually the hit is there. So here all these algorithms and tools in this bioinformatics and this all those approaches in these tools, they're generally based on the probability based approaches or the tools there. So here all the probability, in fact the sequencing approaches are also lies to somewhat to an extent in a probability based approaches. So here the confidence level that the protein that the sequences we are identifying or we are getting, that is it is exactly the same or the thing. So we need to have a confidence level or a confidence score. So the statistically significant we get by the p value or the significance score. So here we try to get through e value or the p value. So I am doing showing a blast. I don't know. I means might be this you will not be able to see, but what results I'll get, I'll try to pick it here. Also, you all can try on your own. So with that accession number, if you just I'll be performing the blast with this only, so you can identify that what are the different types of. I'm pasting it in the chat box, the accession numbers. You all can try and find out that what are those what are the hits you are getting, what is the p value, what is the e value. So it will also help you with uh, in the different assignments solving also that you are. So let me do, and I'll paste the. So this is the sequence. So I'll request you all to perform it, though if you all know also, and try to find out that you are getting the same result or not. So this is the tool. I'm pasting the link here. So I think you all can have got those 4 tabs that I have showed. This nucleotide blast, blast text, T blast 10 and protein blast. So we will go to the nucleotide blast. So here we can see that here you will get that you can enter accession number, GIs or faster sequences any. So here we can retrieve, either we can retrieve the faster sequence like we have done and paste it in the nucleotide blast, or if we know the accession number, we can directly paste the accession number. It will automatically, since it is the same accession number, it will automatically go and I retrieve the sequence. We don't need to do anything. So we will keep the database. So here you can see an organism which is optional. It is given that you can either include any organism. So that will depend upon whether you have been asked to in an or whether you are, if you know, if you have collected something, some organisms DNA sequence, and you know that it belongs to a particular DNA sequence, then it's fine. If it is not, then you can leave it. Or if you want to exclude particular organism, you can play with the different parameters here, and you can look at that what are the different databases with different results you are getting on those changes. And finally, there is a program selection optimized for highly similar sequences, somewhat similar sequences, or more dissimilar sequences. So you can also change the different things. So here I am giving that somewhat similar sequences and perform the blast. And also there are additional algorithm parameters which if you are asked for, or if you want to go for more stringency, you can perform those. So hopefully if you all are doing it, so you all have not got any problem till now, right? So this is, after this we can see that different sequences and the different percentage identity and accession length and what are the top hits we can get. I'll just take a screenshot and paste because it is not visible, it's not getting visible. Hopefully you, if you have done, you are getting this kind of result, right? So here we can see that Homo sapiens mRNA for hemoglobin alpha chain, because we have given that only, so that has been that is quite in a correct way. It has given that it is coming as 100% with a percentage identity of 100%, means it is fully identical. Uh, yeah, so the video, it is being live streamed in YouTube, and the link will be shared with the by the NPTEL team. So this is the sequence per accession length, and this is the accession number. So from here, if you are asked that what are the top hits of the organism, what are the major organisms you are getting identification. So here we can see that we are getting the identity, percentage identity, and here from here only, if you go, we go and click on this accession number, so you can see that the percentage identity is little bit less. So here you can get one or two nucleotides that are different, different, and from here you can identify that, understand that we can get some of the differences between the nucleotides. And also these algorithms are only taken forward where now you can with not this, it's not so simple, but still from the idea of that you can understand that the different tools have been developed as still evolving, where different uh, this SNP, these SNVs are getting and helping us in understanding the things. So now this is what you all can go and look at each accession IDs, what are those organisms, what are those sequences, where is the differences you have found out, and how you can interpret those, or the that you can identify that this is a synonymous mutation or non-synonymous mutation, this is an indel, or that is also important, because from here after this comes, after your alignment to a reference genome comes to identify those differences, how much impactful those differences are. So I'll be sharing, going to that minute, that from here how we will go. So this is just an overview of what is an SNPs are and how it is looked at. So we can see that there are three individuals, they have the similar sequences. I'm saying similar sequences because one individual in the fourth nucleotide position is A, another is G, another is T. So these are the three different. So if we look at the codons, the if it is the genetic level, if it is a three triplet codon gives the one amino acid, so you can check out that whether this changing from A to G is affecting, or A to G is T is affecting or not. So what kind of SNP is it, Is it a missense SNP, or it is a nonsense SNP? Is it uh, forming a stop codon, or that the protein will become truncated or not? So what is an SNP? So SNP, if you look at that, it will contains majority of the sequences similar, but at a single nucleotide level, it will have a difference. So this is how you can identify one SNPs at the genomic level. Also, you can identify that, for instance, here also you can say that GGC, CGC, now it has been changed into, so for this is the complementary sequence. So for in the sense from G, it has changed to A. So this is an example of an SNP where from G to A has been changed in a second individual. So this is how you can identify which one sequence is an SNP. Then you can try to identify with the codon basic sequences that whether it will affect or not at the protein level, at the codon level, or how much it will affect. So next is that, so here this is an example of after getting our genomic sequences, a whole exome sequence, then aligning it to a reference genome, that to identification of SNP. So these are done by a different set of tools which we will come across throughout the whole course, in the coming weeks. So this is just to give you a brief that how much it is important to understand and how we can interpret through these different tools and software, and how we can interpret those data. So see, this is, for example, total 362,000, means 3,62,000 getting total variants are being detected at the whole nucleotide level. So this is the SNP of the 312894 has been identified as an SNPs. So at the single nucleotide level, with the reference in reference to the reference genome, this many has been identified as an SNP. This has been identified as MNPs. This has many as insertions, deletions, and the mixed. So from here, number of effects by impact, how many are having how much impact, the number of count, and the percentage. So we can see that the moderate, high impact, high low impact, and the modifiers, and the number of effects by functional class, that is the missense mutations, nonsense mutations, and the silent mutations. So these are all of the non-synonymous classes. So how much count and how much percent. So this is an example of from where we need to go where, and still this is not the end. From here further we need to identify those at the protein level, and then only we can say that what effect it's actually, theoretically we can say that this is the effect, this should be the effect. So what at practical level or at the phenotypical level what it's affecting, and how we can understand and address those to help treat in those diseases. So this is the type of SNP classes which we need to identify and understand. So for this, I have said that there are many public domain accessible available. So for you all, for you all your curiosity, I have said that there in the SRA sequence read archive, many tools and databases are there. So one of those tool is SRA toolkit, which helps us to fetch the data, retrieve our whole exome sequence or whole genome sequences in FASTQ format. So generally the genomic sequences come in the FASTQ format, from which the downstream analysis are there. The first step what we can do if we are not generating our own data is that for retrieving the public access data. So one of those data is the SRA toolkit. So this is, I am leaving on you all to go and try to read about SRA toolkit, try to find out that how you can install. So this is generally a Linux based tools mostly. So if you all can go and do it in your virtual machine or somewhere, you can try if you all are interested. And really, if we go to this level of data analysis, and also to identify at this level that how genomic changes are affecting at the protein level. So I have left upon you to all to go and read about this SRA toolkit, how you can install this, and how you can retrieve the sequences. The one by one we will try to give you some uh, about those tools that are available, and also to at different free these all these tools are generally freely available. So how we can go ahead and use those tools to finally try to analyze and interpret our data. So if you all have any problem, we can discuss every week. So that will not be an issue. But for your curiosity and your interest, so you all can try and explore this SRA toolkit along with your daily assignments, weekly assignments, and all in the NPTEL lectures, whatever is there. So you all can try. This is not an compulsory or anything, but for your curiosity, and you all can get to know something extra or try to learn also. So this is what for this particular week, where I have discussed about different problems that you can that can help you in solving for this week's assignment, as well as try to give in some flavor of in a case study of what how actually these are all gets reported in the studies, and how actually we can start retrieving the data, and we can get to the different kinds of SNPs and modifiers, and we can understand how they are affecting or creating different diseases. So thank you for this. So and if anyone has any question or any difficulty.
Sir, in the assignments,
Yeah,
I could not get the fourth question.
sorry, if you can tell it little louder. You could not.
I could not get the fourth question.
Fourth question. So I don't know means what questions
In
are.
week, okay.
Yeah. So actually, what we have been asked for is to discuss the types of the problems. So if you can say what type of problem, I can help
Okay,
you.
okay. Which of these quantification techniques is not linked with mass spectroscopy based proteomics? The options is iTrack, SELAC, T4PNK, and TMT.
Okay, okay. So the proteomics in the label-based approaches. So I think it will be discussed later in a detailed manner when the proteomics approaches system. So the proteomics techniques generally can be identified in two ways. One is the label-free techniques quantification, and one is the label-based techniques. Okay. So the label in the label-free technique, there has been no label is being attached to any amino acid sequences. Labels means labels are the tags, or that you particularly make sure that this is the uh, tag, and from where you measure the differences in the control and the disease state. You give one tag in the disease sequence and one tag in the control sequences. So level free doesn't is not equipped with any tags. For the level base, there are three types of labeling techniques that are used. One is the TMT based, and that you say tandem tags. So these all will be uh, said in the detail when you go to the proteomics approaches, proteomics techniques. So that is will be done in the later past. I think it was this question was the week zero, I guess, for the basic thing, and the SILAC is there, and the third option is wrong. What you asked, T4PNK, I guess. So there's not a labeling based techniques in the proteomics approaches. So don't worry about this question. So you will be getting to know about this in the detail in the when the proteomics part will be there. Hopefully.
Okay, sir. So week zero can be submitted at the end, week zero assignment.
I think week zero is not counted for. I'm not sure about this, but I think uh, week zero is just for practice, I guess.
Okay, sir.
Yeah. Any other question or any other doubt from this week one?
Sir, also I did not get one more question from
Yeah.
week zero assignment. Which
Yeah.
of the following databases primarily makes UniProt knowledge base? SwissProt and TRMBL,
Uh,
TREMBL and HMDB, HMDB and HPE,
so
SwissProt and HPE.
so for this, so if you go and search in the Uniprot, you will get to know about it. That it is generally mentions of the Swiss prot and the TRML. So I think this also will be covered in the protein sequences. So if you go and try to read about their info, so generally these are the databases. Uniprot is one of the reference databases for the proteins from where generally like NCBI is for the nucleotide databases. Similarly, Uniprot is for the protein databases, so which is being curated from Swissprot and Tremble databases and has been reposited here. So you can go and read about those databases in detail a little bit if you all are interested. Okay, so here
Okay, sir, thank you.
mostly we'll get to know about all those proteins and proteins what you will try to know about their function. Yeah. So any other question from week one or any other thing from today's session if you all have any query or doubt? Okay, if not so, then I will close the session today and hopefully we'll meet next week at the same time with some for the week two. Also, we will try to, if you all try yesterday toolkit and you can ask that if you faced any difficulty or and also we will go ahead with some next step of how to after retrieving sequences what we can do. So so then we can end this session, right? So okay, so thank you all.
Thank you, sir.